So we now know that the phenotype is the outward appearance of the individual. Now this might be a physical appearance, or it might be some part of a metabolic process. So the inability to break down lactose, or the production of a particular enzyme, such as that fish odor syndrome enzyme that we were talking about. But things aren't always as they appear. So a good example of this is skin coloration. So humans and other animals have a gene that contains information for producing melanin. This is the chemical that's responsible for giving our skin its coloring. So this gene is one of many that influence skin color. But there is, unfortunately, a defective non-functioning version of the melanin gene that can be passed on in particular family lines. So an individual who inherits 2 copies of the defective version doesn't produce the melanin pigment, and they end up with a condition called albinism. So this is a disorder that's characterized by a lack of pigment or very little pigment in the eyes, the hair, and the skin. And so if you have seen pictures of these individuals or have seen individuals with albinism, no matter what skin color they should be, quote, air quotes again, their skin appears really light. Their hair is often light as well. Sometimes their eyes appear red. Because they're not producing the proper pigment. So if you are the parent of one of these individuals, you probably have normal skin color, and it's impossible to tell if a normally pigmented individual carries a defective allele just by looking at them. You could do genetic analysis of them, but just by looking at them, you don't know what their genotype is. So we can look at their phenotype. These are the features that are visible to the eyes. It might be coloration, height, presence of antlers, things like that, or things that are harder to see. So it might be, as I said a few minutes ago, the presence or lack of presence of the lactase enzyme, chemicals that produce blood clots, things like that. And of course, we now know that the underlying phenotype is controlled by a genotype, or the organism's genetic composition. So this is encoded in the gene sequence for a particular allele. And then, of course, we've already gone through the process of gene expression where we actually use the information in the gene to produce a protein. So in our example here, the protein that's being produced is the pigment melanin. So now there's some different possibilities now that we're familiar with a couple of new terms. Individuals who suffer from albinism are homozygous for the defective melanin allele. That means they have 2 copies of the defective allele. Now that other deer in this picture, or the other parent, might be homozygous normal or heterozygous, and there's no way to tell those individuals apart by simply looking at them. But we can use the study of inheritance in order to deduce what the genotype of that individual might be. Now, this is not some super complicated thing to do. We actually have a quick little trick called a Punnett Square. You guys have probably done this if you've had biology in high school. And it's just basically a little diagram of the gametes that an individual produces or possibly produces in order to see the probability that an offspring of this cross is going to have the particular trait that you're looking at. So a Punnett Square is essentially a table of gametes. So that means it's a little diagram that shows what alleles were contained in sperm and egg. So let's look at the 2 giraffes here. We've got an albino mama giraffe up there, and then we have a normally pigmented father giraffe. So let's assume that we know the genotypes here. And actually, we can assume that we know mom's genotype. Because mom has the albino phenotype, and we know that albinism is caused by a recessive allele, we know that she has 2 copies of the albino allele. So that means that she is homozygous. Now, if you remember from our discussion of segregation, that means that all of her eggs are going to contain the recessive albino allele. Remember, if her egg or the cell that's going to produce the egg is homozygous, then each of her alleles are segregated into different eggs, and no matter what, they're going to be the homozygous allele. Now, let's just assume for now that dad is also homozygous. Maybe we know this for sure, maybe we don't. But his alleles are going to segregate through the same process in meiosis, and his sperm are each going to contain a dominant allele, or the normal pigment allele. So inside our Punnett Square is essentially a table showing all possible combinations. Now, I like to think of this as like a multiplication table, and the reason for this, we'll talk about that in just a little bit. But if dad contributes a dominant pigment allele and mom contributes a recessive albino pigment allele, then this individual is potentially going to be heterozygous. Now, he's going to be normally pigmented because he has 1 copy of the dominant allele. And then if we look at all these other guys, no matter what individual we have, dad has to give the normal pigment, mom has to give the recessive. And so we end up with all heterozygous individuals. Now, I want to point out the dominance and recessive references here. If a trait is labeled as recessive, you require 2 copies to see that trait. If the trait is labeled as dominant, it masks the recessive and the heterozygote. So you can have only 1 copy of the dominant allele and still show the characteristic. That's what makes it dominant. So now let's do a different cross here, and let's cross a heterozygous female and a heterozygous male. Now, if you will follow through this table of gametes and perform your Punnett Square, you will see that we have some different combinations from the last cross. So when mom's alleles segregate into her eggs, half of her eggs are going to receive the dominant allele, half of them are going to receive the recessive allele because she's heterozygous. So each of her gametes are going to have a 1/2 chance of receiving either one of those. Same thing for dad. He is heterozygous, so half of his gametes are going to receive the dominant allele and half of his gametes are going to receive the recessive allele. So if the dominant allele from mom and the dominant allele come from dad and fertilization happens, we end up with an individual who is homozygous dominant. That means they have 2 copies of the dominant allele. Let's go down here. If we have the recessive allele from mom, recessive allele from dad, we have homozygous for albinism. So 1 copy of each of the recessive alleles from each parent. Now, there are 2 ways that we can get a heterozygous situation. Either mom can donate the dominant, dad donates the recessive, or dad can donate the dominant, mom can donate the recessive. Either way, we have a heterozygous individual who is displaying the dominant trait. Now, let's apply some Mendel to this. So 1/4 of our offspring are homozygous dominant, and they are normally pigmented. 2/4 or 1/2 are heterozygous. They display normal pigmenting. So here, 3 out of 4 are normally pigmented. And then we have our 1 out of 4 individuals are homozygous recessive. So you can see the 3-to-1 ratio here. I want you to think about what was happening in Mendel's flower experiment. So his F2 generation was the result of crossing heterozygous individuals from the F1 generation. So if we go back here, you can see our true breeding female, our true breeding male, the parental generation. These guys in the center would be the F1 generation. Let's ignore the fact that these guys are brothers and sisters. And if we were to breed any two of these, a male and a female, we now are breeding our F1 generation and producing our F2 generation. So here we see that recessive allele come back where it was hidden in the F1 generation, and now it's back. So that's part of the reason that we can call this recessive. And then of course, because we started with true breeding parents and then we cross the heterozygotes, we can also see our 3-to-1 ratio. Now this is an example of something called a monohybrid cross. A monohybrid cross is when you cross two individuals with respect to one trait. So we're looking at a single gene trait here. So a few minutes ago, or probably in the last video, I said that genetics was really just a big game of chance. So segregation and fertilization are both chance events. If you are heterozygous, you have a 50-50 chance of passing each allele to your gametes. And then fertilization is another quote-unquote random event. And what I mean by that is that you have no control over the sperm or the particular egg from the two individuals that are going to breed. So any one of the millions of sperm that are produced and any one of the thousands of eggs that are produced during a fertilization event can actually contain either one of those alleles. But we can apply some rules of probability to these. Even though this is kind of a game of chance, or a crapshoot, if you will, we can still apply some probability rules and still have the ability to sort of predict how many offspring we would expect based on any particular traits. So I said that if we have mom who is albino, she has a 100% chance of passing on that recessive allele. It doesn't matter which egg gets it. It doesn't matter which egg is fertilized by the sperm. She has a 1 out of 1 chance of passing on that recessive allele. So the probability of her passing on a recessive allele is 1. Now, dad is heterozygous. The probability of him passing on the dominant allele is 50%. The probability of him passing on the recessive allele is 50%. So there's a 50% chance that dad is going to pass on the recessive allele. So the chance that any given offspring, any individual that is a result of this fertilization event is going to be the multiplicand of the probabilities. What that means is we're going to multiply the probability of mom passing on the allele times the probability of dad passing on the allele. So in this case, there is a 50% chance that the offspring will be albino. So let's look at the disease Tay Sachs. So we have talked about this a few times. Now, one really important application of all this is found in the field of genetic counseling. So if two individuals know that they come from a population where Tay Sachs is in a higher frequency, they might want to know what's the chance of us passing this very deadly trait onto our children. And so we will look at something called pedigrees in a few minutes, which is basically a family tree of a genetic trait. But you can use the probability that mom has the allele and the probability that dad has the allele to determine how likely it is that the child will have it. So let's make this easy. Let's say that mom and dad both know that they are heterozygous for the allele. So mom knows based on family history that she's probably heterozygous. That means there's a 50% chance that she's going to produce an egg with that particular Tay Sachs allele. Let's say that dad also knows or has a pretty good idea that he is heterozygous for this allele. There's a 50% chance that he is going to pass on that recessive allele. So to determine the probability that their child could inherit Tay Sachs, we're going to multiply mom's probability, 0.5, times dad's probability, 0.5. So the overall probability that they would have a child with Tay Sachs is 25%, or 1 in 4. So at this point, the family would know that at any given fertilization event, there's a 25% chance that their child is going to inherit Tay Sachs. Now, they might have one child, be totally fine. They might have 3 children, be totally fine. No one's passed on the allele. But the higher number of individuals that you have, the higher number of offspring, the more likely that you're actually going to encounter this chance event. So if they have 4 children, there's a 25% chance that one of them is going to have this allele. Now, that's not to say that their first child might inherit Tay Sachs, because it's a 1 in 4 chance. That means if you flip, just like flipping a coin, there's a 50-50 chance of getting heads. There's a distinct possibility that they would have one child, and this one child would have Tay Sachs. So let's say that you're in charge of alligators at the zoo. Some of your alligators come from a population that is known to occasionally produce albino alligators. Now, none of your current alligators are white, but because white alligators are really popular, you want to increase zoo visitors, and so you want to produce some albino alligators from a controlled mating program. So, the first problem that you're going to have is that you don't know the genotype of your alligators. You know what genotype you want. You want the homozygous recessive in order to get those albino alligators. But the ones that you have don't show the characteristics. So they are either homozygous dominant for the albino allele, or they are heterozygous for the albino allele, meaning they have one normal, one recessive copy. In either case, their phenotype is normal. So determining the particular genotype is your first problem. And this is often a challenge when people are trying to selectively breed organisms. Because the albino gene is invisible, they cannot readily tell who has it. Fortunately, there's a specific tool that we can use called a test cross. When I say tool, I really mean a sort of a process. So a test cross is a systematic way to reveal an unknown genotype. So what you would do is you would borrow, air quotes again, a homozygous albino individual. In this case, let's say we get a homozygous female from another population. Now, if she's if she's albino, you know her genotype. She's homozygous for the recessive phenotype. Now, the catch here is you only get her once. You only get her for one mating event, and you need to figure out what your population is. So let's allow her to breed with one of your males, and you don't know if he's homozygous or heterozygous. In performing a test cross, you are using a genotype that you know and crossing it with one that you don't know. Then you can look at the offspring and say, okay, I got this set of offspring. Therefore, I can work backwards and think about what the genotype of that male was. So let's say that you cross this homozygous white female with a normally pigmented father, and you get all normally pigmented individuals. Now, this strongly suggests that that male was homozygous dominant, because all of their individuals were normally pigmented, which means all of those individuals are now heterozygous. But the key here is that now you know, or have a really good idea of what that generation's genotype probably is. Now let's assume that you have enough individuals in the clutch to rule out chance events that you haven't gotten any white individuals. The other possibility is that the dad is heterozygous. Now, if he's heterozygous, we're going to get a different situation in the offspring, because if dad is heterozygous, half of the offspring are going to be heterozygous because half of his gametes are going to have the dominant allele, and so all of the individuals are going to be normally pigmented. However, half of the individuals are going to get his recessive allele. That means half of the offspring are going to be albino. So now, if this is the scenario, we know what genotype that dad was. We also know what genotype the offspring have to be. So really, there are only two possibilities in a test cross. If one parent is homozygous, the other one is homozygous, then all individuals are going to be heterozygous with the dominant phenotype. The other situation is that half are heterozygous with the normal or dominant phenotype. The other half are going to be the homozygous recessive, or the recessive phenotype. So test cross is really sort of a powerful tool to look at your population and establish the genotypes when they were previously unknown. So let's move on to my favorite part of genetics, and this is pedigree analysis. So let's say you have your group of alligators and you've done some genetic analysis on them, and you want to keep track of the traits, because you don't have an infinite alligator population. Alligators take a long time to breed. They're expensive to keep. So you don't have this sort of unlimited population. But when zoos and things have these controlled breeding programs, they keep track of the pedigrees of their animals. They know who the parents are. They know what traits they have. So a pedigree is a really useful tool to determine whether or not a trait is dominant or recessive, and also whether it is sex-linked, meaning located on the X chromosome, or if it's autosomal. So we'll talk about that a little bit more in a few minutes. Pedigrees are also really powerful tools for humans, because humans are really bad model organisms. We have really long generation times. We have small number of offspring. I mean, even if you think of a family that's a huge family, they really are a small number of offspring compared to some other organisms on Earth. And most families tend to have a small number of individuals anyway. So we're just not good for genetic studies. And that's why the pedigrees are such a powerful tool. So a pedigree is essentially a family tree following a particular trait within that family. So let's talk about some of the things that are going on here. So males are indicated by a square, females like a circle. A line across means a union, whether this is marriage or otherwise. It indicates that there was a union there and they had some children. So the line coming down is the children. The children are listed in birth order. So aunt here was the oldest, uncle was the second, and then this guy labeled father was the third. The same thing over here, sister, oldest, me, apparently, middle, and then sister, youngest. The shaded boxes indicate that they display the trait. So this is not always a bad trait or a mutation or disease. These can be used to follow any particular trait. So it just indicates that they have the trait. And then, of course, they are also listed by generation. So up here on the top, we have the grandparent generation, the parent, aunt, and uncle generation, and then the the generation, the most current generation. So we can use this, we can study the patterns that we see in order to determine the inheritance pattern of our trait. So whether or not it is dominant or recessive. So let's look at a pedigree for something called anury in dogs. So anury is a condition where dogs are born without a tail. Now this is inherited as a recessive trait. So let's look at this pedigree as sort of a puzzle. So let's see if we can determine the genotype of the individual with the question mark. So both of the parents are normal. We know that because they're not shaded. This individual here has the trait. So we start off knowing that this individual is homozygous recessive. We know that this individual doesn't have the trait, and we know that this individual does. So here we know that this individual is also homozygous recessive. So we can work backwards using that information to determine what these parents were. So if these parents produce an individual who is homozygous recessive, that means they each had to contribute a recessive allele to that individual. Great, so I have filled in the genotypes. We can go over this in class if you would like, that's totally fine. But we know that these parents are heterozygous because they produce a homozygous recessive child or puppy. Over here we know that this one is homozygous recessive because they exhibit the recessive allele. Now, we know that this individual does not have this individual does have a tail. That means they don't have the homozygous situation for anury. So we know that this individual has to have at least one dominant allele. So they are either AA or big A, little a. Now, we want to know the chance that the puppy produced from these individuals, that any puppy would also have the anury condition. So we can use the rules of probability to figure this out. So if you, on your own, write out the Punnett Square for this guy, the heterozygous cross here, you will see that there is a 2 out of 3 chance that this individual is going to be heterozygous. Now the reason is that it's 2 out of 3 and not 1/2 is we know that she cannot be homozygous recessive. So we are only looking at the the dominant situations. So ignoring the 1/4 chance that she is homozygous recessive because we know she's not, the chance of her being heterozygous here is 2/3. Then the chance of her passing on that allele for anury is 1/2. So the chance of a puppy being without a tail is going to be 1/2 times 2/3. So there is a 1 in 3 chance that a puppy from that litter would have the anury condition. So as I said, we can go over this in class again. Keep in mind that you can work these out based on the doing the individual Punnett squares. So if it helps you, you might want to pause, do this Punnett square, and then do this Punnett square here to see the overall probabilities, because it's not just the probability of this cross, big A, cross little a, little a. We have to take into account the probability that she is actually heterozygous. Right, so there are some other really cool single gene interactions. Sometimes we don't have a clear case of dominance. So sometimes we have a situation where both of the alleles in a particular genotype can be present in the phenotype. So we're going to talk about these two interesting situations. One is incomplete dominance. And this is where the phenotype of the heterozygote is intermediate between the other two homozygotes. Now, this is not a case of blending. Remember early on in this lecture, we talked about blending where two traits sort of make a halfway point. The idea of blending is that now there is an intermediate allele. In incomplete dominance, both of the alleles are still present and you can get back the original characteristic. So here's an example of white and red flowers. These are snapdragons. They're a pretty little plant. They almost look like orchids. They're multiple flowers on a particular stem. And this is a kind of classic example of incomplete dominance. So let's say that we have the mom who has white flowers and the dad has red flowers. Now you'll probably notice right off the bat that we're using a little bit different notation here. We use this notation because we are indicating that neither of our alleles is completely dominant over the other. So in our previous notations, we were using a capital for the dominant and a lowercase for the recessive, but here we are indicating that this is something different. Neither one is particularly dominant over the other. So mom is CW. The C stands for color. The W stands for white. The dad is CR. So C stands for color. R stands for red. So each of these individuals are homozygous. Now, in incomplete dominance, the heterozygote individual is displaying an intermediate phenotype between the two. So in this case, it's pink. So we can see here we've got CWCR, that's the heterozygote displaying the pink characteristic. So we've got our parental generation, our F1 generation. Now let's cross our F1 individuals and see what happens in the F2. So now we are crossing our heterozygote individuals, two pink flowers, and now we get all kinds of crazy going on here. We get some individuals who are homozygous for white, some individuals who are homozygous for red, and some individuals who maintain that heterozygote pink characteristic. So now we can see both the original parental phenotypes, the white and the red, and we can see that the heterozygote produces that different phenotype. Now you would expect when you cross heterozygotes to get that 3-to-1 ratio, but when you see alterations of those expected ratios, you know that there's something else going on. So instead of a 3-to-1 ratio, we actually see a 1-to-2-to-1 ratio here. So another type of gene interaction is called codominance. So in codominance, we get the distinct appearance of both alleles. So you can kind of think of this as dominant together. Now I really like this picture on the right because it really shows what we're talking about. Now these shirts are obviously not a genetically inherited trait, but you get the idea. Dad is wearing horizontal stripes. Mom is wearing vertical stripes. The child here has both horizontal and vertical. It's a really fantastic representation of codominance. So in the real genetic world, an example of this are what we call checkered chickens. So this happens when you cross a white chicken with a black chicken and you get black and white chickens. Again, our notation's a little bit different here because the alleles are behaving differently. Neither one is dominant over the other. We get dominance at the same time. Actually, let me go back. I forgot to talk about an example of incomplete dominance. So I don't know if you remember, but back in chapter 4, we talked about endocytosis, and there's a specific type of endocytosis called receptor-mediated endocytosis. Now, in one particular example, these receptors on the outside of the cell bind to cholesterol, and then through the process of endocytosis bring the cholesterol into the cell. Now, there is a genetic predisposition for high cholesterol, and this is called familial high cholesterolemia. It's a really big word to say that you don't have the receptors on your cell to bring cholesterol into the cell. Now remember that cholesterol is actually important. You do need it. But individuals who suffer from this disorder cannot bring cholesterol into the cell. Therefore, they accumulate cholesterol in the body, and this leads to heart disease. Now, high cholesterolemia is a really good example of incomplete dominance. Individuals who are homozygous normal have all the receptors they need, no effects of the high cholesterol. Individuals who are homozygous recessive have no receptors at all, and so they can't bring any cholesterol in. This is often fatal by the early 20s because they just have so many heart issues associated with this. Individuals who are heterozygous actually produce receptors from the normal allele and no receptors from the other allele. And so you get sort of a halfway amount of receptors. You have half the number of receptors. So the disease is sort of intermediate. You can have some problems, but they're not as severe as with high cholesterol. All right, so we are moving into sort of the interactions of multiple alleles. We talked about incomplete and codominance. Now let's talk about what happens when you have more than 2 alleles. So everything we've talked about thus far has been 2 alleles, purple and white, or normal pigment, abnormal pigment. But there are certain situations where, and actually many genes, display multiple alleles. Now the interesting thing with multiple alleles is that you can have normal dominance, you can have incomplete dominance, or you can have codominance, and that can differ depending on the alleles that you are looking at. So the most classic example of this is our blood type. So our blood type, our A, B, and O blood types, are actually made up of 3 alleles. We have the A allele, the B allele. Both of those are dominant to the O allele. So here we've got the indication of the recessive with the lowercase. And you might notice that we've got that different notation with the superscript here. Now that indicates that together A and B are codominant. So this can be quite confusing. If you were to write out all of the genotype combinations, you could be homozygous for A, which produces the A blood type, or heterozygous for A. Remember, A is dominant over the little I, or the O allele. And so those guys are both going to show the A blood types. The same situation happens with B. B can be homozygous or heterozygous dominant over O, and you get the B blood type. You can also have a codominant situation where you have A and B together. This is the AB blood type. And finally, you can be recessive for both alleles and be the O blood type. Now these types refer to a particular type of receptor on the outside of the cell called antigens. Now, when you have the A allele, you produce A antigens on your blood type. When you have the B allele, you produce the B antigens. If you are O, you produce neither A or B antigens, and your cell is sort of naked on the outside, I guess you could say. So if you have 1 copy of the A allele, you're going to produce A antigens, 2 copies A antigens. O allele, no copies. If you are codominant with the AB blood type, you have both antigens on your blood. So there you can see you are displaying both characteristics of the both dominant characteristics. You've got your codominance here. So this is important if you are talking about receiving or donating blood, because if you are type A, I don't mean personality, I mean blood type, type A blood, your body produces basically an immune response to type B blood. So they produce these little antibodies that recognize B blood and would attack it if you were to receive B blood. So A type cannot receive B. B produces B produces A antibodies. So if you were to receive A blood, your B blood is anti-A and would attack it. A and B blood type produce neither antibodies, and this makes sense because they have both receptors on their cells, and so you don't want it attacking your own blood. So there are no antibodies. There's no immune response elicited in response to receiving either A or B. Type O is the opposite. It has neither antigen, but it produces both antibodies. So if you were to give this individual A or B blood, or even AB blood, you are going to get an immune response where the body doesn't like the A and the B antigens. So there's quite a complex science behind blood donation, all related to having these multiple alleles and this codominant situation. So this is just another summary chart showing the antigens and the antibodies. This is kind of showing what the genotypes would look like. So individuals who are type O, who are not producing or who do not have any antigens on the outside, are considered universal donors. They can give to anybody because those blood cells are not going to be attacked by their immune system. A and B blood types, because they don't have any antibodies, are universal recipients. They can receive blood from anybody. Now, B can receive B, can also receive O. A can receive A, can also receive O. So it is quite complicated. If you're interested in this, you can Google it and get a little bit more information. So there's one more marker on our blood that has to do, that affects our blood compatibility. Now, this is called the Rh factor, and this is responsible for the positive and the negative. So you might be O negative or A positive in your blood type if you know it. The Rh markers operate under a single gene with only 2 alleles. So this is not a multiple allelism example, but it is a second gene that affects our blood type. So the Rh positive is dominant over the negative. So individuals who are Rh positive are either homozygous positive or heterozygous. Individuals who are Rh negative are homozygous for the negative. Now, an individual who is positive can receive positive or negative blood, but an individual who is negative cannot receive positive blood. So you can see how on top of the blood types, this also complicates things a little bit.